Sherbrooke, and he'll be talking about RNA-seq methodology, methodological landscape, the ignored importance of the choice of genome annotations.
Hi. Oh, yeah. Thank you, Michelle, for the introduction, and I'd like also to thank the COSI for the opportunity to talk here. So today I want to put forward the idea of community oversight in RNA-seq and also highlight the importance of genome annotation in RNA-seq quantification. So RNA-seq is a quickly evolving method, and the average dataset has quite changed throughout the years. And to ensure a sound processing of an RNA-seq dataset, research groups must stay ahead with the ever-updating state-of-the-art technology. And by looking at what the community is doing, we might identify important actions that need to be taken to ensure that all data is rightfully treated. With this idea in mind, I went through a methodological review of the RNA-seq literature, mainly going through the literature and looking at what tools were being used and how they were being used. And I will only present really a high-level summary of this study right here. So the first key point is that the RNA-seq methodological literature is incomplete. Not all the papers specify all the relevant information that has been used to generate a RNA-seq dataset. We can also see that some steps are better specified than others, mainly alignment and quantification, while others are worse, like pre-processing and annotation. But the key question is, does it really matter? If annotation and pre-processing do not have any impact on the final quantification, then it would be a loss of time and effort to try to rise these numbers. But if they do have an impact, then we should worry about these low numbers and we should do something about it. We also have to keep in mind that RNA-seq methodologies are quite heterogeneous. There's a wide number of different tools and references that can be used for each of the different steps. Well, in every step, the vast majority can be explained by only a handful of tools, but they still have a long tail of different software that is being used and that might induce some biases in the literature. And if one wants to create a new RNA-seq pipeline, one way of doing so would be to go through the literature, looking at what kind of benchmarking we can find and use the benchmarking to identify the right choices of tools. But to do so also requires having an idea of the relative importance of each of the different steps. And here I took in the several articles that I have found that are analyzing more than one step of RNA-seq pipeline at once. And mainly we can see that they only consider alignment and quantification as being part of the RNA-seq workflows, whereas annotation only have one articles that have used more than one. And these annotations have only been used to identify transcript assemblies, and they have not been compared in quantification. So we currently lack a meaningful comparison of a workflow-wide RNA-seq benchmarking. And with this idea in mind, I went with a permutational study of RNA-seq workflows, mainly selecting a few human healthy tissues and some of the highly used tools I found in the literature and using two different versions of Ensemble. You'll see why later on. And I've computed every possible permutation of tools there is in this matrix, which gave me a pretty big count matrix. And to analyze this data, I've used an independent component analysis, also known as ICA, which is a dimensional reduction techniques, which lets me draw sources out of this data set. Sources can be think of as gene expression patterns, where each source has a number of significant genes either in blue, which means that they are lowly expressed, or either in red, which means that they are highly expressed in the source. And this is only a small number, there are many more. And by looking at one permutation at a time, we can compute a score identifying if the source is present in the permutation, so we can identify what common gene patterns can we find in every possible permutation. They can either score very high, medium, or low. And by computing all the different scores of all the different sources with all of the different permutations, we can actually classify the sources by the information they give us. So for this example, this source here scores very low for every tissue, except for the data sets that are coming from the heart. So we can say that using this source, we can identify if the dataset was a heart or not. And by combining all these possible different sources, we can identify the tissue of origin. But the sources that are interesting to us are these ones. These ones are sources that actually correlates with technical steps in RNA-seq, mainly the annotation, the alignment, and the quantification. Oh, well, this label is not supposed to be there. And for the remainder of the presentation, we'll be interested in this one. So we can see that we have two different sources that each learn onto from different gene patterns how to differentiate between ensemble and ref-seq. So that means that we have genes which are impacted by the annotation and that we can use these genes to infer which annotation has been used. And we also have this one which lets us differentiate between ensemble 92 and ref-seq versus ensemble 96. And we'll be interested in this one because it's an easier story to draw because we only have to look at what are the differences between ensemble 92 and ensemble version 96 to learn about the differences. And the idea was to find a common feature that all the gene in the annotation, all the genes that change between the annotation share. And well, you can't see it right now, but it's a percentage of overlapping locus. So in ensemble 92, all the genes that are present in the source actually have a very low percentage of other genes overlapping them, whereas in ensemble 96, nearly all these genes got new overlapping genes over them. And this has impacted the quantification. Just for a quick idea of what does a new overlapping looks like. So this is the annotation in ensemble 92. So we have a gene and another gene, which is SRSF1, and we have this transcript, which is overlapping only a small part. And this is a new isoform in ensemble 96, which now overlaps a very important parts of SRSF1. And this is impacting SRSF1 quantification, and SRSF1 is a highly studied splicing factor. We can also have novel genes, which have been added, which acts some kind of a read-through, which now overlaps the entirety of two different genes. And we have a third example where we have some genes that have been split up into different genes. So here AS and SD1 has been split up into its main isoforms, its upstream ORF isoform, and a coding sequence. So far, we've seen that genome annotations are badly reported in literature, and that genome annotations are ignored in workflow benchmarking. But that really matters because genome annotation actually induce biases in the RNA-seq quantification. But wait, there's more. That's only a small part. That was only one of the different sources, and from each of the different sources, we can learn a different story highlighting a certain hypothesis that was disregarded by a certain tool or considered differently by other tools. So I won't have time to go through all of these, but there are more interesting stories to draw and other conclusions that will help us create better tools and create more informed tools. I'd just like to acknowledge my group, my supervisors, Michel and Ryan, in the research group, also ISMB, Eclipse, and the RNA Cozy for the travel fellowship, and open source tools. And I also like to point out that I'll be finishing my master's degree by the end of December, so I'm currently looking for a PhD position if anyone's interested. Thank you.